Good morning, everyone. I think we can get started. So thank you for attending our cardiovascular grand rounds. And it's our great pleasure to invite Dr. Marlene Rabinovitch to speak at our cardiovascular grand rounds. Marlene received her MD from McGill University and pursued a postdoctoral research in Harvard Medical School in pediatric cardiology. And currently, she holds the Dwight and Vera Dunleavy Professorship in Pediatric Cardiology at Stanford University. And she is also the Director of Basic Science and Engineering Program, Betty Irene Moore Children's Heart Center at Stanford University. Marlene is a great leader in investigating mechanism and diagnosis and treatment of pulmonary hypertension, and also a leader in elastin biology. Our group is very interested in elastin patient signaling pathway, and also using elastin to enhance engineered patient blood vessels. So we have great interests in Marlene's research. So we are very happy to invite Marlene to be here. And Marlene's interesting study led to the pulmonary hypertension diagnosis and a new treatment, and published multiple exciting papers in circulation journal. Her group also made seminal discoveries about neutrophil elastase contribution in the loss of precapillary vessel, and also the proliferation of small cells that occlude the pulmonary artery, and eventually leading to a pulmonary hypertension. And her group discovered the elastase inhibitor, Elastin, and this is currently in clinical phase 1 trial, and also resulted in multiple exciting papers in Nature Medicine, JCI, and JCB. And recently, Marlene's group has utilized the high-throughput small molecule drug screening strategy and bioinformatics strategy to discover novel therapies that can enhance BMPR2 function. And they discovered that the immunosuppressor FK506 can enhance the BMP function. And this exciting research is currently in phase 2 clinical trial. And they pushed this program and combine the BMPR2 and the PPAR gamma axis, and studying the mitochondrial function during pulmonary hypertension. And this work has been published in Cell Metabolism, JCI, and JCB. Marlene's research has been funded by NIH R1 program and PO1 program. And she was a distinguished scientist lecturer at the American Heart Association, and she won the Robert Group Prize at the American Thoracic Society. And she is an excellent mentor and won the multiple mentor awards of excellence in Stanford University, and Judy's Poole Mentoring Award of American Women in Science. And Marlene has showed great leadership in science by serving as associate editor of Circulation Research and editor of Annual Review of Physiology. And she is serving on multiple scientific board for NIH, American Heart Association, and Max Planck Institute. So it's great to invite Marlene to share with us her exciting talk, the molecular mechanism inform new therapies for pulmonary hypertension. So Marlene, welcome.
Thank you. Thank you very much for that kind introduction. Can everybody hear me? Good. Okay. So I'm absolutely delighted to be here. Thanks, Yibing, for the invitation, and Eric. It's a pleasure always to see old friends and to meet new ones, and especially to meet new young colleagues excited and interested in medicine and science and how you kind of merge the two to find new and better treatments for patients. So I'm going to start with an introduction to pulmonary hypertension, and then I'm going to tell you where we are with the studies that we're doing and how we think that those studies are really informing new therapies that we hope will be disease modifying. So pulmonary hypertension, when I got into it, of course, is a major complication of children with congenital heart disease. At the time that we started, we didn't realize which children had reversible pathology and which children had irreversible pathology. But some of the studies we did actually led to better monitoring of these children, earlier diagnosis, and certainly earlier surgical treatment, which has been a major and important change in the field. But we still see children that have advanced disease, even with modest congenital heart defects. And now we're seeing a new burgeoning group of patients that have stiff vessels as a result of single ventricle physiology and non-pulsatile flow. In adult cardiology, there's a growth industry of HERVK, or heart failure with preserved ejection fraction, that's also complicated by pulmonary hypertension. We see it now increasing in patients with metabolic syndrome. In fact, women with pulmonary hypertension have a very high incidence of metabolic syndrome. In lung disorders, not surprisingly, there's pulmonary hypertension, particularly in children that have developmental abnormalities of the lung. Sickle cell disease is associated with pulmonary hypertension that is a high risk of morbidity and mortality, independent of the fact that the pulmonary pressures may not be very high in that disease. So it still is an area of active investigation, particularly by colleagues such as Mark Gladwin at University of Pittsburgh. California, we're seeing a great deal of pulmonary hypertension associated with substance abuse, primarily methamphetamines. And worldwide, it's a complication of HIV and schistosomiasis. Even in cases where schistosomiasis has been eradicated, pulmonary hypertension now presents as a lethal complication. Autoimmune disorders, particularly scleroderma, but not infrequently rheumatoid arthritis and even lupus, are seen as associated risk factors for pulmonary hypertension, and liver disease with portal hypertension. But the interesting feature is that the incidence and severity of pulmonary vascular disease in all these primary disorders is probably governed also by genetic factors and genetic predispositions. So you can have children with congenital heart defects with huge shunts that don't develop severe vascular pathology, and other children with very modest shunts that appear to. And this is some of the area that we're trying to investigate and trying to better understand using patient-specific cells, which we'll get to a little bit later. Now, the other form of pulmonary arterial hypertension is more rare. It's idiopathic. There's no known etiology. There is a mutation associated with familial forms, which I'll discuss in a moment. It affects females more than males. The ratio of 2 to 1 is modest. In some series, it's as high as 4 to 1. It's usually diagnosed in the 3rd to 4th decade of life, postpartum, but we see it in infancy and in childhood in our clinics. And as I mentioned, it's rare. 15% are familial, which means that there's more than one affected family member. And of those cases, 70% have a mutation in a receptor, bone morphogenetic protein receptor 2, BMPR2, which is a receptor in the TGF-beta superfamily of receptors on the cell surface. Now, 20% of patients with non-familial idiopathic pulmonary hypertension also have a mutation in this receptor. But it's very curious that if you look at family members that have the mutation but are unaffected, they account for 80% of individuals. So the penetrance of the mutation causing the disease is quite low. And that suggests that there are other very important modifying factors. Now, over the years, investigators like Wendy Chung have found other genes that are associated with the development of familial forms of pulmonary hypertension, but they seem to converge also on this receptor pathway by and large. So ALK1, for example, is a co-receptor of BMPR2. SMAD9 is a signaling molecule. Caveolin-1 interacts with a BMPR2 receptor. The potassium channel crosstalks with it, and EIF24 alpha kinase is a direct target. TBX4 and SOX17 are transcription factors that may be co-regulated with BMPR2. So we focus on this receptor because in patients with all forms of pulmonary hypertension, even associated forms, and also in experimental models, there's reduced expression or function of this receptor pathway. redirecting us to try to understand why having a normal pathway is protective of the pulmonary circulation. So why is this important? Well, our current therapies really are not doing the job. They're primarily vasodilator therapies. They were used in a heroic manner initially in patients that weren't even responding by vasodilation to these agents, but over time it was shown that they not only improve quality of life, but also survival. Whether they work on the pulmonary vessels that are still somewhat open and not completely shut down, we don't know whether they work better in earlier forms of the disease or whether they even just work on the coronary circulation is still open debate. But the three forms are intravenous or prostacyclin or other analogs that can be given subcutaneously or now even by oral or inhaled forms. Endothelin receptor blockers that are potent anti-vasoconstrictive agents, and phosphodiesterase inhibitors that impact the nitric oxide synthase pathway and increase nitric oxide availability, all aim to try to open up what's not completely closed or shut down. They improve, as I mentioned, quality of life. But on the other hand, the progressive obliterative lesions that I'll show you in a moment in the pulmonary arteries dictate that eventually patients succumb to heart failure and the need for heart transplantation. So this is the armamentarium that many of our patients need to assemble every day for subcutaneous treprostinil or intravenous therapies. And it does mean that it does impact their quality of life, but it's far improved over not having these therapies. And there's just a little guy with a backpack that our patients carry around all the time. Some of them are also on supplemental oxygen. So we've done much better than previously. It was usually a 2-year death sentence before these therapies. Now we're seeing a 65% 5-year survival, as shown by these curves, but we can do better than this. And that's the aim of my research program as well as others over the country and internationally. So to do better, we really need to address the underlying vascular pathology. So if you look at the pulmonary circulation shown by this angiogram, you can see marked differences compared to a control pulmonary angiogram. Namely, there's rarefaction of the distal precapillary arteries, so you don't see them as a background blush. And the more proximal arteries are narrowed all the way up to the hilum. So the loss of the small vessels is in part due to programmed cell death of the endothelial cells. They're very vulnerable to injury, and they just die and take with them pericytes and even smooth muscle cells, or they obliterate and close down completely and then resorb. And then the narrowing of the more proximal arteries is due to an exuberant proliferation of cells that Dan Greif studies that are smooth muscle-like. His fate mapping shows that they can originate in experimental models from the muscular media. There's also some evidence that they can originate as a form of endothelial to mesenchymal transition. So from endothelial cells, evidence by Kurt Stenmark suggests that the fibroblasts can become myofibroblasts and can contribute to the neointima. And Dan's also shown that inflammatory cells can become smooth muscle cells when they're recruited to the vessel wall. And inflammation is part and parcel of this disease, and we'll discuss that a little bit later. So the question is, how do we explain this vascular pathology and how do we tackle it? And in particular, how do we explain its relationship to the BMPR2 signaling pathway? So over the years, we established how this pathway is important in protecting the pulmonary circulation and in preventing the development of these lesions. So we showed that BMPR2 through PPAR gamma promotes regeneration of the endothelium when it's injured. We showed also that PPAR gamma downstream of BMPR2 can suppress smooth muscle cell proliferation when growth factors are released, again, when the vascular bed is injured. And we also showed that BMPR2 through a different pathway can suppress the recruitment of inflammatory cells. So this pathway is very important in maintaining vascular homeostasis. It's also important in the assembly of elastin fibers. In fact, the major factor that initiates the formation of the elastin fiber, TGF-beta, requires BMPR2 in order to translate elastin into protein and allow it to assemble on top of a fibrillin scaffold that BMPR2 also regulates. And then most recently, we've shown that when cells undergo DNA damage, either genomic DNA or mitochondrial DNA, BMP through PPAR gamma is also important in the repair of that DNA damage. So this is what BMPR2 does. What happens when you don't have BMPR2? And not only do you lose these protective effects, but the problem is you also gain adverse pathology. So we showed that if you lose BMPR2, there's endothelial to mesenchymal transition. So what happens is the endothelial cells undergo apoptosis or programmed cell death, illustrated here, but the remaining cells, in order to hold on and to survive, undergo a transition to a smooth muscle-like cell. That doesn't mean they become that exuberant neointima, but that means that they no longer can control the smooth muscle cells that are there to proliferate. And so what happens when endothelial cells become smooth muscle cells is they can't suppress them anymore. But in addition, the tight barrier that endothelial cells form is lost because mesenchymal cells are looser. So substances and particular inflammatory cells get into the vessel wall that are normally excluded. And also the endothelial cells are potent anti-inflammatory cells, and they lose that capacity as well. They don't produce the molecules that are there to fight inflammation and to protect the rest of the vessel. And this work was done and published a few years ago by Rachel Hopper in Circulation. And Rachel identified the molecular mechanism whereby this transition occurs. She showed that there's a chromatin remodeling factor that is critical in initiating transcription or gene regulation of a transforming factor called SNAIL. We also know from our work and other people's work that in addition to loss of BMPR2, other factors can co-conspire in causing this abnormality, such as disturbed flow at bifurcations, oxidant injury, chronic inflammation, and even DNA damage can contribute to this transformative process that impacts the vessel wall. The other feature that is changed when you lose BMPR2 adversely is that the increase in the proliferation of smooth muscle cells takes place spontaneously. It doesn't require even growth factors, and that's due to an upregulation of an enzyme called aldehyde H1A3, which was also discovered on RNA sequencing as being one of the most highly upregulated genes. So we thought, is this enzyme important in changing metabolism or in changing the regulation of genes that are absolutely critical to cells that need to proliferate in response to loss of BMPR2? And we worked out the signaling mechanism whereby the loss of the receptor leads to the upregulation of this enzyme. But we really, and the work is still unpublished. It's by Dan Lee, and I'll give you a few examples of how this works. But before we pursued it from our sequencing studies, we wanted to be sure that this enzyme was highly expressed in the vessel wall of patients with pulmonary arterial hypertension. So aldehyde H1A3 is red, smooth muscle actin is green, and the enzyme is in the nucleus, and you can see it by this pink staining overlying the blue staining of the nuclei in patients with this exuberant neointimal formation, but not in the control vessel wall, shown here with the thin muscular media. So we said it's there, we're on the right track, what is it doing? And so we knocked it down to be sure that it was critical to proliferation of the cells, and in fact, when we did that, using RNA interference technology, we could slow down the proliferation of the smooth muscle cells to the level of the controls from the patient. And we also slightly decreased the proliferative capacity in response to serum of the control cells. So then we had to figure out whether it was important pathologically in the context of disease. So we knocked it out in smooth muscle cells in mice. And what we found was that the animals that are exposed to hypoxia develop pulmonary hypertension, the normal animals, as assessed by right ventricular systolic pressure or right ventricular hypertrophy. But if you knock out this enzyme, shown here in green, in the smooth muscle cells, the animals don't develop pulmonary hypertension. It's not significantly higher than the room-air controls, indicating that it was necessary for the development of the disease, and it's necessary for the muscularization of the distal vessels in the mouse in hypoxia. So if you look at a normal mouse after a few weeks, 3 weeks of hypoxia, you can see this dense brown muscularity of small vessels and also larger vessels. But if you look at the knockout, the mouse that doesn't have this enzyme, the vessels remain relatively non-muscularized, and the quantitation is evident as well. So how does this work? Well, what we showed is the enzymes in the nucleus. One of the products of the enzyme is acetyl-CoA, and we showed that acetyl-CoA acetylates histones, and when it does that, this epigenetic feature is critical in opening up chromatin. So how does it know what chromatin to open? Well, in response to loss of BMPR2 through the same signaling factor, there's coordinate upregulation of a transcription factor, NFY. So at sites where NFY binds, the chromatin is opened up, and genes that are important in proliferation are all upregulated. And those include genes that are important in energy metabolism that the cell needs, particularly glycolytic metabolism, nucleotide synthesis, purine pyrimidine production, and also cell cycle. So this enzyme is actually the fuel for the whole process, and it's downstream of loss of BMPR2. It goes up to induce this exuberant response. So I've shown you that the receptor is important in endothelial cell homeostasis. I've shown you that it's important in preventing proliferation, and I've shown you also that when it's absent, these factors seem to go rogue. So the inflammatory response also requires input from BMPR2 to dampen it down when a vascular bed is injured. And we showed this when we knocked down BMPR2 in endothelial cells. One of the key factors that recruits monocytes and neutrophils is GM-CSF. And rather than a slight it's a little more than a slight increase, you get a twofold higher upregulation of this very potent chemoattractant when you lose BMPR2. And the process we identified is due to a perturbed ER stress response due to an increase in translation of the cytokine from the messenger RNA work published about 5 years ago by Dr. Sawada. So GM-CSF is there, and it recruits cells with a receptor for GM-CSF, like mononuclear cells or macrophages. So here you can see them expressing the receptor for GM-CSF in the neointima. And here you know there are monocytes because they express a marker CD68 for monocytes. So now you have these cells drawn in. They're drawn in through the endothelium, but also through the adventitia. So is GM-CSF important pathologically? And there's more and more evidence now from Kurt Stenmark and other investigators in different models that it is. But if you give GM-CSF, you can make pulmonary hypertension worse in mice. So the RVSP goes up a little bit higher than it does normally. But the important feature is that if you block it with a neutralizing antibody, you can prevent the recruitment of monocytes and the pulmonary hypertension. So here you can see a little bit in brown that macrophage staining occurs in hypoxia, is exaggerated with GM-CSF, but is ablated if you block it with a neutralizing antibody. So then we said, okay, well, you have a more exuberant inflammatory response, but we see the inflammation smoldering in patients with pulmonary hypertension. So we asked, what is it that perpetuates this response? Why doesn't it just happen and go away, as you might expect? So the clue to why this might be happening was really in work that was done by Professor Marc Humbert and his colleagues in Paris, where he showed that adjacent to vessels that were occluded or partially occluded, there were these little tertiary lymphoid nodules that had all the markers of a lymph node. And so we wondered, what were they recognizing in the vessel wall? And he had also shown that there are autoantibodies that are circulating, but they were circulating autoantibodies that weren't very informative of the pathophysiology. So we thought if we could figure out what they were recognizing in the vessel wall, that might also give us a clue to the disease pathology, but also to why it was being perpetuated. So what was done and published a couple of years ago by Toshi Sato and J.R. Moonen from my lab is that by isolating the immune complexes in patients with pulmonary hypertension, we were able to identify antigens that were highly prevalent in the tissues of our patients that weren't there in immune complexes that were far less prevalent, but that we could isolate from control unused donor lungs. And by recognizing what these antigens were by isolating them from the immune complexes using mass spec, we thought we would have an inroad into pathophysiology, particularly if these antigens recognized key proteins in the vessel wall. So we did that with the help of Bill Robinson, and we found that in the complexes, there was increased expression in the patients of an antiviral protein that, to the date that we were studying it, was really only very well studied and reported as an anti-HIV protein, so preventing a replication of HIV. But we knew that our patients didn't have HIV, they were screened for it, so we wondered if it was in response to another virus. So you can see lots of SAM-HD1 complexes in our patients compared to the donor lungs, and so we then began to look to see where this protein was being produced as an antiviral response, and what was the virus it was recognizing. So here you see a stain for endothelial cells in red, and green is the antiviral protein. So it's there in endothelial cells. It's there in macrophages. This is a stain also in red for macrophages. Here it is. It's there in antigen-presenting dendritic cells, not so much in T cells. So we said, okay, what is the antibody in the tertiary lymph node recognizing this antigen? And sure enough, it is. When we overlay the protein, we pick up the IgG from the tertiary lymph nodes. So they're making the antibody to SAM-HD1. So it looked like a good fit. And then, but it still was questionable. What was it doing there? Did it have another function, or was there another virus that it was recognizing that was latent? So we collaborated with Charles Hsu at UCSF, who had engineered an approach to identifying the virome in lung tissue. And sure enough, we didn't see any of the known exogenous viruses like flu or paraflu, but we did see increased expression in our patients, at least in most of them, of these endogenous retroviral elements. And we expanded our group and sure enough picked up expansion of these endogenous retroviral elements in the tissue of our patients, both the messenger RNA, the genomic DNA, as well as the protein products. So what are these endogenous retroviruses? Well, they're sequences that are stored in all our genome. They're remnants of ancestral retroviral infection. They're not infectious, but they can produce products when cells are under attack to induce an innate immune response. So they have been identified as amplified both in cancer and autoimmunity, but without really understanding how they're produced, how they're amplified. It's thought that in most cells the DNA is methylated, so they're shut down, but they can be demethylated and actually expressed in tissues. For example, in stem cells, in embryonic stem cells, these elements are upregulated. It's thought to induce an innate immune response to protect the stem cells, work of Joanna Wasoka at our institution. But then when the cells differentiate, they become methylated and they're shut down. But for some reason in our patients, they're becoming amplified and upregulated. So which cells are expressing them? It appears to be the myeloid cells, the mononuclear cells. Here's an antibody stain where you can recognize the HERVK product, the DUTPase, which is an antiviral protein that induces an innate immune response, as I'll show you. And here we showed that when we take the recombinant form of this protein, we can actually induce the antiviral protein, SAM-HD1. So it produces protein products that are very potent. The protein products are highly expressed in monocytes, circulating monocytes from patients with pulmonary hypertension. The monocytes then get into the vessel wall. It's expressed in iPS cells, so even in induced pluripotent stem cells from our patients. It's expressed, the product can activate B cells, accounting for the fact that the B cells are producing antibodies to the antiviral protein. And it also increases cytokine production, both in endothelial cells, shown here. HERVK can stimulate IL-6, also TNF-alpha, and in monocytes, it can stimulate IL-6 and TNF-alpha, even in low doses. So does it induce pulmonary vascular disease? Is it enough to do that, necessary or sufficient? So we injected animals with 3 doses weekly of this DUTPase, and sure enough, they developed some pulmonary hypertension. In some of the animals, we injured the endothelium, but we really didn't need to do that to bring out a phenotype. And in fact, we were able to show that in response to this product of the retrovirus, they developed increased muscularity of their pulmonary arteries. So how does this all work? Well, it works in concert. Why it's upregulated or demethylated, we're really not sure. And what its relationship to loss of BMPR2 is, we're also not sure. And it could be an important modifier of the disease, accounting for the fact that some patients with a mutation develop disease and others don't. But the idea is that it is increased predominantly in myeloid cells. We're trying to understand why this demethylating enzyme is suppressed or increased in myeloid cells. The DOTPase, the product, is released. Inflammatory cells become activated, as do endothelial cells. I'll show you in a moment that it also produces elastase and neutrophils. It produces antiviral proteins. And by activating B cells, it produces the antibody to the antiviral proteins. And as a result, immune complexes are formed. And subsequent to that, complement is activated. And both the activation of complement and the inflammatory cytokines can co-conspire, as can the products of HERVK, to induce pulmonary vascular remodeling. In recent studies, we've shown that the product of HERVK can also induce endothelial to mesenchymal transformation through a toll-like receptor. So it seems to be a very important co-regulator of the disease pathogenesis. So then we said myeloid cells, I wonder if this is also upregulating in neutrophils, and I wonder if the neutrophils are also abnormal in this disease. So why did we start to think so? Well, when we measure circulating neutrophil elastase, it doesn't matter what the etiology of pulmonary hypertension is, all patients seem to have elevated levels. So the patients with idiopathic disease, portopulmonary hypertension, congenital heart disease, elastase is elevated. So since neutrophil elastase are the big producers of circulating elastase, we look to see whether or not the neutrophils are producing the elastase. But why do we focus on elastase, and why have I worked on it for so long? Well, when I started to look at lung biopsies from children with congenital heart disease by ultrastructure, one of the most striking observations was degradation of elastin. Now, if you look at more advanced lesions, you can certainly see it. Elastin is the black material that bounds the muscular media of the vessel wall, the internal elastic lamina next to the endothelium, the external elastic lamina next to the adventitia, and a thicker vessel has more elastic lamina, like an elastic artery. But now you see in an occluded vessel, it's totally disintegrated, even by light microscopy. There are fragmentations, duplications, there's loss here. And this is important because this increases vascular stiffness mechanically. You get an abnormal collagen to elastin ratio. Bob Meekum has shown that when an animal loses elastin, it dies in the early postnatal days because of proliferative changes in the coronary arteries that become totally obliterated. So it controls smooth muscle cell proliferation. We have multiple levels of control in the vessel wall. It actually sustains the microcirculation. So if you lose elastin, you lose distal vessels. And the breakdown products of elastin, Bob Sr. showed, are highly pro-inflammatory. So you need elastin for multiple reasons in the vessel wall. So sure enough, for whatever reason, these neutrophils produce more elastase. Same number of neutrophils. If you measure elastase protein by a Western immunoblot, they have 3-4 times more. They're the same size, so they're just packing it up. And it's biologically active. If you stimulate the cells, they produce more active enzymes, shown in green, compared to the control cells that release elastase at the same time. Moreover, it's been shown that when neutrophils become angry, they release nets. It's thought to be a very important mechanism in trapping bacteria, but that process also requires elastase. So you can see that we've stimulated control neutrophils, and they're wimpy. They extrude a little bit of chromatin, but if you take the pH neutrophils, they just explode. And in fact, it's often hard to isolate pH neutrophils and try and do anything with them because you don't have to do very much for them to extrude chromatin. And they do so by an elastase-mediated mechanism, and what happens is the elastase traffics to the nucleus, breaks the nuclear membrane, the chromatin is extruded. That traps bacteria if the bacteria are there, but it also causes a lot of collateral damage because the elastase is now released and the endothelial cells become impacted by it. Also shown recently in pulmonary hypertension that there are a lot of nets in the vessel wall, work of Martin Wilkins. So if you block elastase, you can actually prevent the net formation or attenuate it down to the level of the controls in the pH cells, suggesting that it is an elastase-mediated phenomenon. Okay, so we asked whether these neutrophils were abnormal in other ways that would make them pathologic, and we measured simple things like did they adhere more to a substrate or to endothelial cells, and sure enough, they're much more tightly adherent. And then we said, well, do they migrate faster or slower? And in fact, when we look at transendothelial migration using a well where endothelial cells are on the top and the neutrophils are added, and then we see what goes to the bottom in response to a cytokine, you can see that they don't migrate very well at all. When you look at pH endothelium, it's leaky, so normal neutrophils go through, but these cells kind of get stuck. They get stuck because they're more adherent, they're less migratory, and their speed of migration is also slowed down. So we said, well, why do they have this property? And that would make them release nets and be more pathologic within the tissue. And so we did a proteomic analysis trying to find out if we could figure out what proteins are responsible for this impaired adhesion. And the one central node we identified that could be responsible for the increased adhesion that downregulates molecules like the beta-2 integrin that are important in migration is vinculin. And it's been shown that vinculin forms focal adhesion contacts that attach the neutrophils very tightly to cell surfaces or substrata. So we said vinculin, how is this all tied together? How is it tied to elastase? So we did observe, and this wasn't our work, but previously by the work of Brown et al., that vinculin is also an antiviral protein. It's part of an innate immune response. So when you stimulate cells with HIV, vinculin goes up to actually decrease proliferation of the virus and actually decreases viral entry as well. So we thought that's very interesting. It's an antiviral protein. Maybe we should circle back to the HERVK. And indeed, when we looked at transcriptomic analysis, most of the genes that surfaced as being highly upregulated in our neutrophils were part of an antiviral interferon response. So we said, okay, is HERVK elevated also in the neutrophils? And sure enough, like other myeloid cells, it is. So this just shows you the messenger RNA for these retroviral elements, both the envelope protein messenger RNA, or the envelope mRNA for the protein, and also the HERVK DUTPase. So, okay, is this all part of the same story? So we wanted to make sure that the protein was there. So this is confocal microscopy, and you could see the HERVK protein in the patient neutrophils, not in the controls. And then we said, okay, well, the protein is there as a protein product secreted, and sure enough, it is. And the protein product of these retroviruses can upregulate vinculin as part of that innate antiviral response, and it can also upregulate neutrophil elastase in a neutrophil cell line, suggesting too that neutrophil elastase is part of this innate immune response. So the cells are toxic, but they're toxic because they feel they're under siege by a viral protein all the time because of this expansion of these retroviral elements. So let me go back. So now I've shown you another element where you're enhancing recruitment, not only of mononuclear cells, but of neutrophils that are highly toxic. And I also indicated that you have impaired assembly of elastic fibers in patients with pulmonary arterial hypertension, and that creates a perfect storm. Because if BMPR2 is necessary to assemble elastin, and the elastin is flimsy, and you have high levels of elastase, that causes more degradation and more release of elastin peptides and more smooth muscle proliferation. So here's an example of control cells, smooth muscle cells we use, or fibroblasts shown here. And when you put them in culture and stimulate them with BMP or TGF-beta, they produce elastin fibers shown by this green mesh material and quantified here on the right. In patients with idiopathic pulmonary arterial hypertension, the elastin is rather flimsy, not well associated or hung together at all. And if you have a mutation, it's even worse. You get very little assembly of elastin in response to these factors. So it looks like, and we were able to document this in animals that lack BMPR2, that the elastin is not well assembled. The fenestrations of the elastin are very large. This work of Nancy Tojeb published a couple of years ago. So the ideal pulmonary arterial hypertensive therapy then should be required to activate BMPR2 and to suppress inflammation. And so we looked to see what therapies might do that. And in fact, the study that Yibing had alluded to was a study in which we carried out high throughput analysis of the response of cells in terms of turning on BMPR2 signaling to 4,500 either FDA-approved compounds or compounds that could be repurposed that were in development. And the top of the line, our top hit, was actually the immunosuppressive tacrolimus. So here's an immunosuppressive that also activates BMPR2, and I'll show you how in a moment. And then a little later, we did a study looking at elefen, and we observed that elefen had anti-elastase properties, but also seemed to preserve endothelial homeostasis. And we then showed that, by serendipity, elefen, which is an elastase inhibitor and an anti-inflammatory molecule, can also upregulate BMPR2 signaling. So how does this work? Normally, when BMPR2 signals, the ligand binds, and a break on the ligand called FKBP12 is moved away, and a phosphatase that degrades the signal is also moved away. So now the secondary or the co-receptor becomes phosphorylated, signals and genes become expressed that protect the cells. If you have loss of the receptor or wimpy signal because you have haploinsufficiency, so our patients only have one allele that's mutant. They have some BMPR2, but usually about half the amount that they need. They need a little bit of help. So what FK does is it binds that break, FKBP12, and it also blocks the phosphatase. So it pulls it off the co-receptor. And as a result, a wimpy signal becomes a more prominent signal. Now, elefen, that's an elastase inhibitor, works a little bit differently. Elefen binds to caveoli, the circular molecules, in these deep crevices. And in so doing, it holds the receptor there longer and prolongs the weak signal. So both could work in concert, and both seem to work very similarly in augmenting BMPR2 signaling, but also in suppressing the inflammatory response. So is there another strategy that we can use to augment BMPR2 signaling? And this is just recent work in our lab where, again, by accident, we found in looking for genes that were upregulated in response to hypoxia in endothelial cells, we found BMPR2 is upregulated transiently, but it's there, as I'll show you in a moment. And what we showed was that this increase in this receptor is important in causing cycling of endocytosis, so the receptor is internalized. It then forms into multivesicular bodies that are extruded as exosomes that are laden with BMPR2. It's almost like a self-preservation of a cell that's under attack to let the BMPR2 out of the cell so it can protect other cells and amplify the signal. So that's transient hypoxia that you might have when you have a sleep apneic episode. But if you don't have BMPR2 to do this, your cells are not protected. So here's an exosome. That's what it looks like. They're small particles. They can be measured, so we know we're isolating them. And they're augmented in hypoxia. That just isn't our work. Other people have identified that. What we've identified is that BMPR2 is critical for the release of these extracellular vesicles. So you lose BMPR2 in cells and you lose the burst of extracellular vesicle production. You increase BMPR2 with chloroquine, you can increase extracellular vesicle production, but if you knock down BMPR2, the response becomes attenuated. And BMPR2 is augmented in these exosomes. You can see it. They're just containing this packaging material. They even have the message we've been able to identify. So it protects cells. If you take pulmonary hypertensive endothelial cells from patients with a mutation and you give them the extracellular vesicles from a normal patient, they can form nice tubes so the endothelial cells regenerate normally. And also if you take smooth muscle cells and you add the extracellular vesicles, the smooth muscle cells take them up, and that dampens the proliferative response. If you expose those smooth muscle cells to serum, the extracellular vesicles keep them from proliferating as robustly. So it has a profound protective effect. So if you make induced pluripotent stem cells and you you correct the mutation, they can generate vesicles that can actually be protective and can be given to a patient with the disease. So we tested that. The extracellular vesicles, shown in red, are actually taken up by endothelial cells. That's in cell culture. But they're also by PET scanning taken up in the lung if you give a tail vein injection, shown here using gallium-68. And you take an animal that is a wild-type animal, you expose that animal to short bursts of hypoxia, the animal doesn't develop pulmonary hypertension. You lack BMPR2 in endothelial cells, and it does in green. You give it exosomes from normal cells with BMPR2, and the pressure doesn't go up above control levels. But now if you decrease BMPR2 in the exosomes, it drifts back up, even the same number of exosomes. So BMPR2 is important for the generation of BMPR2 in exosomes, but also for the number of exosomes that are released, all of which appears to protect the vessel wall. And during these short bursts of hypoxia, during prolonged hypoxia, it's different. So as I mentioned, the next goal of the study is to try to get iPS cells that are corrected to produce exosomes so we can protect the vessel wall. So the first thing was to go back and make sure that the iPS-derived endothelial cells made from skin were similar to the normal endothelial cells from the same patients or controls. So here, endothelial cells harvested from the lung at transplant from a patient with pulmonary hypertension. Here are the iPS endothelial cells, and they don't form tubes. But you give them elephant, and most of them will start to form tubes. Same cells that form tubes with elephant seem to also be forming nice tubes with FK506, with tacrolimus. So then we said, okay, is there anything else out there we should have on our back burner that we could use as screening tools, not just looking at BMPR2, but looking at restoration of cell health? So this was work of Min-Sha Gu. And as I said, thanks to the surgeons who take the fibroblasts from the skin biopsy when they open up the chest to remove the lungs, we're able to generate iPS cells that we now can differentiate into endothelial cells and smooth muscle cells. We take the endothelial cells first, and we screen them through 4,500 different compounds to see if the endothelial cells survive better when you withdraw serum as a measure of decreased vulnerability in response to one of these drugs. And we look for an appreciable increase in survival. We make sure that the drugs aren't toxic, they're not killing the cells. And then we further screen them to make sure the drugs can help the endothelial cells regenerate or form tubes, and also then that the drugs suppress proliferation. So there's attrition at each step. Some of the compounds go away. But one of them worked across all 7 cell lines that we tested, and that's a tyrosine kinase inhibitor. So you can see cell survival is improved in a dose-dependent manner because an enzyme that's activated when cells die, caspase 3, is reduced. These cells are under serum withdrawal. And then we look at smooth muscle proliferation, and again, the proliferative response is downregulated in response to this tyrosine kinase inhibitor. So that's good. But then are there other compounds that might behave the same way? Because tyrosine kinase inhibitors are going to be hard to get back into the clinic because imatinib, which was used, Gleevec, did not have a very good success rate, had some side effects. So we use a bioinformatic approach that Yibing described previously to try to find new compounds. So how does this work? Well, we take our BAIT compound, our tyrosine kinase inhibitor that worked across all our cell lines. And we say, what is this compound doing across many cell lines? What's its profile? So we look at the LINCS database that looked at this compound across 100 cell lines. And we say, okay, what's common? What cells are downregulated in response to this agent? What cells are upregulated? And that's our BAIT signature. So then we say, okay, do we have a pH signature? Because the BAIT signature should antagonize the pH signature. So this was work in collaboration with Pravesh Khatri at the computer science department. So pH cells have been studied both by microarrays and by transcriptomics, and there's a lot of data out there in geo and databases. So we actually were able to develop a pH signature. And then from the pH signature, we develop an anti-signature. So these are genes that go up in pulmonary hypertension. These are genes that go down, and this is the anti-signature. So now we want to match our bait signature with our anti-signature to find out which gene regulation is really important in compounds that should attack the abnormal gene expression in pulmonary hypertension. So that's how it's refined. We try and match it as close as we can, and then we try and say, okay, does that tell us how it's acting? What genes that are up in pulmonary hypertension, is it down-regulating? What genes that are down, is it up-regulating? Is that informing us? So we find the pathway, and then we say, okay, is there anything else that has the same or even a better signature that's out there? So this is a purely bioinformatic approach. So let me show you in the last slide how it works. So this is the pH signature. This is just an example, down-regulated. This is up-regulated. This is the anti-signature, so we're looking to match the red and the blue. This is the TKI. There's blue in this category, and there's some red all across. This is imatinib. It's a little bit weaker than the TKI, perhaps accounting for the fact that we found a better response to this tyrosine kinase inhibitor. But then we said, what else is there? And here's some agents. Here's a potassium channel opener with even a better anti-signature. So that gives us new opportunities to explore other agents using a bioinformatic approach. But then there's an added advantage because we said, okay, are there any genes that match, any compounds that match the signature? And here we see dasatinib, which is a tyrosine kinase inhibitor, but dasatinib has actually been shown to cause pulmonary hypertension. Sure enough, it matches the red and the blue. And then there are other compounds like a PPI that you might want to avoid because that too has a pulmonary hypertension signature. So you can go the whole gamut from using cell biology, animal models, trying to exploit a pathway that we know is genetically important, trying to find out what other modifiers are there in inflammation that may co-conspire with it, and then try and use bioinformatic approaches to keep the pipeline going to find new compounds. So in summary, I've shown that reduced BMPR2 can induce adverse effects related to pulmonary hypertension, the transformation of endothelial to mesenchymal-like cells, the impaired regeneration of the endothelium in response to injury, the proliferation of smooth muscle cells in response to metabolic changes that impact gene regulation, the recruitment of elastin-degrading neutrophils and mononuclear cells that are also highly activated, and impaired elastin fiber assembly. And therapies that restore BMPR2 function then have promise in reversing pulmonary hypertension. So lastly, I'd just like to acknowledge our funding agencies, Oliver Weedo, who generates the elephant, the recombinant protein that we use in our studies, Maria Ritze, who generates the recombinant form of the HERVK, retroviral gene product, Charles, who helped us with the viral studies, and then multiple collaborators at Stanford, Joe for the iPSCs, Mike for the genomics, Pervesh for the bioinformatics, and then Roham for the clinical input, and as well as Mark Nichols and Bill Robinson for the immunology, and then Joanna for help with the HERVK. Thanks very much.
Open for questions.
So Marlene, that's a tremendous tour de force. Congratulations. I have a couple of questions, or somewhat tangential, but you got me thinking. So I had spent a fair bit of time in Puno at 12,000 feet, looking at individuals who were exposed to indoor air pollution. And I guess the take home from that a couple of year experience was it's remarkably little PAH. And I guess I'm curious in this work, in translating this work, particularly as it relates to BMPR2, if you've looked at populations who are exposed to potential hits like chronic hypoxia and indoor pollution, and have you been able to expose groups of patients who may have different gene expression?
I think that's an interesting observation because I would wonder if it's different in patients that live at lower altitude, because maybe there's a survival factor for people at high altitude and they're not getting pulmonary hypertension, because experimentally you can induce it with pollutants. You can induce it with house dust mites. There's a very pronounced model. It was an asthma model that actually has profound pulmonary vascular changes that are studied. So I wonder if those patients actually have a survival advantage. In fact, children that are born at high altitude that have big ventricular septal defects and high pulmonary vascular resistance actually do much better better than children at low altitude with the same level of resistance. Is it due to vasoconstriction, or is there some survival advantage that you have because you've survived to high altitude?
So
So
related question, I guess,
yeah.
in my mind at least, is with this data that now has emerged around BMPR2, and as we learn more and more about the potential for oxygen toxicity, do you envision scenarios where we might, in fact, genotype patients who are at risk and mitigate and modify their oxygen? We basically give everyone oxygen, right? But some people may have actually may suppress BMPR2 activity, as you suggested, and it might be actually particularly more toxic in those patients. We might let them live more hypoxic. Any thoughts on that?
Yeah, and I think it's the reoxygenation that's much more detrimental than the hypoxia. So, for example, in our studies, when we expose loss of BMPR2 cells to hypoxia, they're not generating exo. It's true, but when we re-expose them to room air is when they get into trouble. So our phenotypes are much more profound because they don't reverse. So normally you take a mouse or a rat, expose it to 3 weeks hypoxia, put it back in room air, everything goes away. Not if you lose BMPR2 either in endothelial cells or smooth muscle cells. So I think it's the same thing with sleep apnea. I think it's the reoxygenation that's more problematic, and maybe because it's a double hit for the same reason. But the mitochondria undergo fission. It has severe metabolic consequences, not during hypoxia, as much as that change from hypoxia to reoxygenation. So I think we don't pay as much attention to oxygen toxicity as we should. Yep.
Hey, Marlene, welcome to Yale. Good to see you again.
Hi,
Two quick questions.
Sorry.
One related to the science. The exosome-derived BMPR2, have you seen that restores BMP response in those endothelial cells?
Yeah, we haven't looked at signaling yet, but we should. But it definitely is there, and it does see. But it is a good question. We really need to see whether it's working through the same pathway, or is the exosome carrying something else with it in addition to BMPR2 that's actually having that beneficial effect. What we know is when we lose BMPR2, we lose the advantage of that exosome, but it could be that BMPR2 is also responsible for producing something else in the particle that's also protective. So we should at least show the signaling is there. Yeah.
And a second, more of a speculative question. Of the therapeutic targets that's originated from your lab that's now in human trials, elephant, FK, and other agents, if you were to pick one that you think has the most promise, which one would that be?
Well, I like elephant, but we don't know. So far, it's in the middle of the phase 1 trial, no adverse effects, thank goodness. So we'll see when we get it to the phase 2, because it has multiple anti-inflammatory effects. It's an NF-kappa B inhibitor. So I think it doesn't have the same immunosuppressive properties as FK. People have asked me, maybe you should just combine them, and that would give us some IP, because we don't have any IP on repurposed drugs. So that's another thought as well. I think that might be too big a hit, and we need to study them separately anyway. But I don't know, and we don't know, no matter how hard we work at this, what's going to work and what's going to fail, absolutely. So that's why we're trying to keep the pipeline going and keep training people who are going to carry it on and like you, and figure it out. Yep.
Can you comment on organ specificity of BMP signal in BMPR2, both in pathogenesis, but also as a therapeutic target? Calcification, other side effects of the treatment.
Yeah, you worry about that because BMP in other tissues certainly is important in calcification. So the idea is to tweak a deficient receptor rather than to augment something, but the site specificity is always going to be a question. Would you get ectopic bone formation? Because I think it's going to be limited. That's why I don't like the ligand. My colleagues, Nick Morrell, is using BMP9 as a therapeutic agent. I worry that is going to be important in ectopic calcification, more than just tweaking the receptor, because ultimately it's going to depend on how much ligand is available. But it is a concern, for sure.
It's a great talk, amazing work. In the metabolic syndrome, obesity syndrome, sort of the rising wave of pulmonary hypertension. Do you think, it's probably multifactorial, but do you think your metabolic mechanism driving acetyl-CoA, perhaps coupled with mitochondrial dysfunction and the shunting into this acetylation pathway, do you think that's a potential mechanism in those patients?
Yeah, I think so. I think so. You're talking about the ages, rage, that kind of pathway that you get into, the acetylated glycation end products possibly as funneling in.
Separate pathway. I'm thinking more of your acetyl-CoA and your
Oh,
acetylation pathway that you talked about.
Yeah. I think so, but then you need the transcription factors. So I think you need two things. So there are many patients with metabolic syndrome that won't get pH. But I think if you have that additional vulnerability and you tell the acetyl-CoA where to go, that's possible that you're going to get it to the targets that are going to cause this exuberant proliferation. But it all is target specific in endothelial cells, not signaling also acetylates, chromatin at sites, but they're sites that improve endothelial regeneration. So depending where you're acetylating, which transcription factor guides you either to turning on an adverse response or a beneficial response.
Okay, so is that question? Okay. Back here, and so let's thank Marlene for this wonderful talk.